Okay, boa tarde, good afternoon, everyone. It is my great pleasure to introduce our keynote speaker for today's keynote lecture of the fourth workshop in Inovação em Engenharia Biomédica, Professor Shankar Subramaniam from University of California, San Diego. I've been knowing Dr. Shankar for a while during my past at UCSD. Dr. Shankar is a full professor at UCSD and has a long story of several appointments and several roles that are of paramount importance for the recent advancement of systems biology, computational biology, bioinformatics. He used also to be president of IEEE Engineering, Medicine, and Biology Society for the years 2019 and 2020. And he has received multiple awards from NIH and from other funding agencies, and is currently an overseas advisor for the Department of Biotechnology of the Government of India, a member of the European Science Foundation panel, and serves on several national research advisory councils, including, of course, the National Institutes of Health. As I said, Dr. Subramaniam is a pioneer in systems biology research, and I'm sure that his talk will give us today a very good overview of several of the applications of his research to multiple fields of medicine. He has published in leading journals such as Nature, Cells, journals of the science family. He was a recipient in the past of the Faculty Excellence in Research Award at UCSD. And in general, we really look forward to your talk, Shankar, and let me also thank you once again for your very kind acceptance of our invitation. We look forward to your talk because you will give us a great presentation of how biomedical engineering can definitely contribute to change in medicine and contributing in general to innovation, which is the common theme, the denominator of this workshop. So I will quickly switch to my co-chair, Dr. Tatiana, for technical communication in Portuguese before we switch back to English and we give Shankar the word for his talk, okay?
Thank you, Federico. Hi, Dr. Shankar. Thank you very much, and I feel very honored to have you here in our workshop. It's a great opportunity, so thank you very much about this. We are sure that we are going to hear a lot of an interesting point of view about biomedical engineering, and we would like to say some words in Portuguese, so I'd like to ask you excuse me. And boa tarde aos presentes. Nós estamos dando início, então, à nossa palestra de abertura do workshop em Inovação em Engenharia Biomédica. Sejam todos bem-vindos. Hoje, nessa palestra de abertura, nós temos o Dr. Shankar Subramaniam. E o Dr. Shankar, ele se colocou à disposição para responder as perguntas durante, inclusive, a sua apresentação. Se vocês tiverem perguntas, vocês podem mandando pelo chat do YouTube, e as perguntas podem ser feitas em português ou podem ser feitas diretamente em inglês, e nós repassaremos as perguntas ao Dr. Shankar. Então, gostaríamos de agradecer a todos e desejar uma boa sessão. E agora nós vamos passar a palavra de volta ao Dr. Shankar. Dr. Shankar, thank you very much, and now the word is yours. Thank you.
Thank you so much, Tatiana and Federico. It is really a pleasure. I'm going to share my screen. It is a pleasure and a privilege to really be joining you in the beautiful afternoon in Sao Paulo, I guess. And next time I want to do it in person because I want to be present to see your beautiful city and your institution. Let me see if I can share my screen for just a second. It says I'm presenting my screen. I will can you see it, Tatiana?
Yes.
Okay, thank you very much. So what I will do today is to describe to you research that goes on in my lab at the interface of engineering data sciences, and pathology. I mean, I'm going to explain to you in just a minute the types of things we do and the opportunities research at the interface presents for all of us. And I want to request you, please feel free to stop me at any time with questions, so I can try my best to answer your questions right away. So you don't need to wait till the very end. I don't mind being interrupted with questions. I sometimes tend to speak fast. I will try my best to control. It's, the Greek story of Demosthenes, who put pebbles in his mouth and tried to learn to speak slowly. So I'm going to try to learn to speak slow. And I will explain to you things that we do in my lab. So my lab is first, before you do anything else, it is customary in the US and in the West to acknowledge all the scientific funding that is provided for research in my lab. And I'm very fortunate to have funding from NIH, from so many different institutes in NIH. And I'm going to tell you about each of these areas. To summarize what we do in my lab, what we do is we use all this combination of methodologies that I'm going to describe to you for looking at finding markers of disease, deciphering mechanisms of disease, for reconstructing networks and building systems models of normal and diseased function from many, many measurements across scales of genotypes and phenotypes in humans. So this is really what we do across all these different diseases. And I'm going to give you a few examples today, which will tell you the kinds of things we can do with such an approach. In the next slide, I'm listing for you publications in the last 4 years from my lab in each of these areas. And I'm going to give you first a high-level view of each of these areas. And then I will go into very specific details into neuropathologies, into Alzheimer's disease, and in COVID-19 immune system, and where we have published recently a lot of papers. And so I'll walk you through these two examples, and if time permits, maybe through a very complex muscle disease called muscular dystrophy. I may not have time for that. So let me just tell you, in the area of neuropathologies, in the last 5 years, we've been very, very interested in trying to understand what is the cause of Alzheimer's disease. And many of you know, as people get older, they get Alzheimer's disease. Sometimes even young people have Alzheimer's disease. This is a disease where people become demented, they lose control of their functions, their brain atrophies, and eventually they die. And there is no medication, there is no drugs, there is no treatment, there is nothing. We even only diagnose Alzheimer's when it is too late. We diagnose somebody as Alzheimer's when they have first dementia, when they keep forgetting things, then they have cognitive function decline. Then they do imaging, and then they find the brain to be full of plaques and tangles. And so that's when you know that you have Alzheimer's disease. And there's no cure. I mean, in fact, only recently, a couple of months ago, there's a drug that is approved, but FDA, the person who in FDA who was the head of the approval body, he resigned saying that it is not a good drug. And so this is really the state of affairs in Alzheimer's disease today. We also work on, I'll tell you a little bit about it in more detail in just a minute. We work on cancer, different aspects of cancer. We work on neuroblastoma, glioblastoma, and I may have time to tell you very briefly about it. We work on colorectal cancer, where we have published some papers on looking at right and left colon. We look at children's liver and look at hepatoblastoma. And then more recently, we have started working very heavily on breast cancer, specifically triple-negative breast cancer in women that has no cure, or that recurs again and again. And so that's an area I don't have time to tell you about today, but maybe at a future time when I visit you, I will give you a lot more detailed story about how we deal with cancer. We work a lot on liver functions and pathologies, an area that's become very, very interesting. And one area where we have worked on in liver is a very rare disease called biliary atresia, where the bile duct does not form in children developmentally. As a result, the liver cannot get rid of its toxic substances, so the liver fails in children. So they replace the liver, and then after that, again, it fails. So therefore, this is really lifestyle becomes very weak for children. So for years, we've been working on children's liver And we have tried to identify the mechanisms by which the liver function can be addressed in hepatoblastoma and in biliary atresia. Unfortunately, I don't have time to tell you a story about this. Again, when I come to your institution, I can give a number of lectures where I can talk about this in great detail. We also work on stress and vascular system over the last many years. I mean, maybe 8-10 years. And so we've really looked at how blood flow, and with Dr. Federico Aletti is very familiar with this area, how blood flow is altered in the case of occluded blood vessels, which means people get atherosclerotic plaques, and so blood does not flow equally well. And so we investigated this to understand what is the consequence and what are the mechanisms that contribute to atherosclerosis. We have published many papers in this area recently to show how we can deal with, addressing the vascular flow system in human blood vessels like arteries and veins. In the striated muscle area, for again many years, we have been working on muscle pathologies. We've been really interested in looking at what causes muscular dystrophy and ALS, which is also known as Lou Gehrig disease. And so we've been looking at mechanisms of how these can be addressed. Recently, we did a very exciting thing which was published in Science Advances a few months ago, and where we showed that how to create a muscle from stem cells ex vivo in the most efficient manner. Is really an engineering problem, and I'll tell you more about it if I have time. The immune system has been of interest to my lab for the last 20 years, 25 years, and we worked on innate immunity, we work on macrophages and macrophage biology. We have published 70, 80 papers in this area. Most recently, we have really picked up last year on COVID-19. As you know, this is a big pandemic, and as a country, as you know, US, Brazil are two of the most affected countries. And so we have been working on COVID-19. We have discovered really fundamental mechanisms that are involved in severe COVID-19 patients. And I hope to tell you about it because this is a very exciting story. We published a paper in Cell, in Nature Medicine. And then just last week, we had a paper out in Frontiers in Immunology, which talks about the complete remodeling of the immune system in severe patients in COVID-19. I will not tell you about these two areas. For many years, we've been working on metabolomics and also on various aspects of computational systems biology. In fact, Dr. Aletti knows that we have developed a large number of methodologies for looking at systems biology using computational methods. Most importantly, we are interested in looking at causality, which means you want to know cause-effect relationships. We are also interested in looking at using deep learning and machine learning methods to understand more biological mechanisms. So these are areas that are active in my laboratory. I will not have time to tell you about any of these areas. I hope to focus only on these areas on the left here, neuropathologies, immune system, and muscle pathologies, if time permits. So I will tell you in more detail about this. Let me first introduce you to this disease called Alzheimer's disease. This is the 6th leading cause of death in the United States and in the world. 5.8 million Americans are living with Alzheimer's disease. 1 in 10 people age of 65 and older have AD. In 2020, AD-related dementia cost $305 billion. By 2050, it is projected to cost $1.1 trillion. Almost 2/3 of Americans with Alzheimer's disease are women, and older African Americans are twice as likely to have AD than Caucasians. I have not looked at Brazil to see what the demographics are, but I suspect that you have a significant amount of AD in elderly people. After investments of tens of billions of dollars in research and development and testing, there is no cure for AD. So to address this question, so years ago, about 5-6 years ago, I was discussing with my colleagues in UCSD who work on Alzheimer's disease, and I asked them, why are there no easy ways of looking at it, Alzheimer's disease? Here are the problems. Number one, there are no good model systems. After all, you cannot take a human brain and cut open the human brain and then look at the brain as Alzheimer's or not. You cannot do invasive way of looking at the brain. You can only do outside imaging, and by the time the imaging shows any defects, it's too late. The second, so we don't have good model systems, and mouse and rat and even primates are not good model systems. Mouse never develops the kind of symptoms or kind of disease pathological phenotypes that humans develop. So how can we develop good models? So that's the first question. The second question that comes up is that, okay, let's say we have, we want to detect Alzheimer's disease early enough so that if you find it early enough, you can find a way of addressing it. How would you find it early enough? Unfortunately, you cannot invade the brain. In the blood, there is no easy way to do it because it's a blood-brain barrier, and there is no easy markers in blood that tells you you have Alzheimer's disease. There are many other diseases which have markers in blood, but not Alzheimer's disease that easily. So that's the second problem. What's the third problem? The third problem is if you find a drug, let's say you find a drug, the drug has to cross the blood-brain barrier. And the drug has to cross the barrier because if it doesn't cross the barrier, it will not reach the brain. You have to find a drug that affects all the disease pathologies. At the same time, it has to cross the brain. So these are all very complex issues. So we said, let us bring some engineering approaches towards dealing with the issue of developing models, trying to find the mechanisms, trying to find drugs which pass the blood-brain barrier, trying to ask the question, will this drug work in a human being or not work, without actually testing it on humans. To test it on humans, you really need to have prior trials. And unfortunately, prior trials in animal models are not useful. So this is the challenge that we are faced with. So to address this question, we took a systems approach to Alzheimer's disease. I have a fantastic postdoc in my lab, Andrew Caldwell. And Andrew really took the job of trying to ask the question, we published 2 papers. This is already published, not accepted. This is an old slide that I'm using, and this is also accepted. So we have 2 papers which now show the hallmark of things. And I'm going to tell you more in detail about it. How did we think about the model system? Before I tell you that, this is a healthy brain in humans, and this is the Alzheimer's brain. What happens in the AD brain? So you have plaques, you have tangles, and these are kind of aggregates of amyloid beta peptides, and these amyloids essentially take over the brain. They break up synaptic connections, and they form these things called tangles, which are essentially deceased neurons, coming from deceased neurons, and they essentially lead to dementia and eventually to cell death and to death of the individual. This is a canonical thing that happens in Alzheimer's brain. So to address this issue, we took the following systems approach. First, we asked the question, can we take induced pluripotent stem cells from deceased patients and then transform these induced pluripotent stem cells into neurons? If you can convert this into neurons, how do you get pluripotent stem cells? We can use fibroblasts, which is skin, and then we can convert the skin cells into induced pluripotent stem cells. And these stem cells can be converted into neurons. We can then characterize these neurons between normal, non-demented control patients and deceased patients. So we can ask the question, what is it in deceased patients that's different compared to normal patients? So there's a lot of engineering that goes on here in terms of trying to do sample preparation, trying to do lineage specification. I won't have time to go into details because we published all of this. And then you look at what are the transcriptional changes that happen in the Alzheimer's brain. Then we look at the gene programs that are altered. We build all of this into mechanistic framework. Then we design drugs which will address these issues. We then take the drug-treated brain models, and then we look at how they differentially behave compared to deceased brain and to normal brain. So this is the program which we developed. I meant experimental program and methodology we developed to address Alzheimer's disease. Do you have any questions for me at this point? I want to pause for a minute and ask you if you have any questions. So this is the basic idea that we went with in order to ask the question, can we address Alzheimer's disease? Do you have any questions for me at this time?
I think not yet, Dr. Shankar.
Not yet. Okay. Not hearing. Good. Not hearing anybody. I'm going to continue telling you. So this, in order to address this problem, we took early onset familial Alzheimer's disease. Now I'm going to tell you a little bit about this disease. There are some genetic mutations. In fact, one major mutation, genetic mutation family comes from South America. And these mutations, for example, in only 3 different proteins, presenilin 1 or presenilin 2, or this protein called APP, when you have a mutation, this mutation or this or this or this or any of these, all the family members who get this mutation develop Alzheimer's dementia and Alzheimer's disease. And this is a young, early age disease. They develop in the age group of 45, 50, 55, etc. It is not something that happens when you're 85 or 90 or 70, 80. It happens when you're 45, 50. And these mutations are homozygous, which means they happen in both alleles. And they are absolutely, I'm sorry, they're heterozygous. If they're homozygous, they're lethal, embryonic lethal. And if this mutation occurs in one of the alleles, then it's not a question of if the person will get Alzheimer's disease. These are 100% penetrant, which means the person will get Alzheimer's disease. It's a question of when they will get Alzheimer's disease. So these are well-characterized mutations, not in my lab, but in other labs. What is this protein presenilin 1? Presenilin 1 is the catalytic subunit of this complex called gamma secretase. Gamma secretase cleaves a peptide called APP. Normal cleavage, all of us, I mean, you, me, everybody else, it cleaves in all the cells, including in neurons. And the normal cleavage gives you peptides which are processed, proteolyzed, and gotten rid of. This is a standard biology in what happens in normal patients. When there's a mutation in this gamma secretase, presenilin 1 unit in the gamma secretase complex, it then cleaves the peptide in the wrong place. Instead of cleaving it in the regular place of the A beta, this peptide called A beta in the position 38, 36, 38, etc. It cleaves it in the position 40 and 42. And the moment it cleaves it in 40 and 42, it forms fibrils. These peptides form fibrils, which means they form associated aggregates, and they make up the plaques that we see in AD Brits. This is known now for last, 30 years or 40 years, 30 years or 40 years, we've known this, that this is what happens in Alzheimer's brains. In fact, there's even a Nobel Prize for somebody who showed that there are formation of plaques, not only in this, but in other diseases as well. Okay, so how do we deal with this issue? So to do this, we started with the fibroblasts, which is skin cells. We programmed them into pluripotent stem cells. And these pluripotent stem cells, we reprogrammed them into neurons or glial cells. This is all very good or straightforward technology, not invented in my lab, but this exists in literature. In fact, you can do it, we can do it. It is very good technology. It's just that you need to be very careful in following the protocol to make sure that you get neurons and not other cells. So we get 90% neurons and a few glial cells. So we took all these familial mutations, and then we took non-demented control, which means patients who are very old, 85 years old, don't have dementia, don't have mutations, and who don't have any Alzheimer's symptoms. So that we can compare against the most worst case scenario of best patients. So then we can ask, how are they different from the Alzheimer's patients? So then we took all these cells, we sequenced and analyzed them. We did RNA sequence. I'm going to tell you in a minute. We did RNA sequencing, ATAC sequencing, and chip sequencing. All of these methodologies are, again, high-end methodologies. In fact, we invented a methodology for RNA sequencing. I may not have time to tell you about it in detail. So the RNA sequencing idea is that you just take the chromosome, and then you get the mRNA, you amplify it, and then you sequence it to see what transcripts are expressed. That's an RNA sequencing method. And you know that companies like Illumina have a spectacular method for doing high throughput RNA sequencing. So you can get tens of thousands of genes, transcripts read at the same time using RNA synthesis, hybridization method, synthesis by hybridization method. And then there are methods like, DNA-seq, FAIR-seq, ATAC-seq, 4C, high-C. We use many of these ChIP-seq. We use many of these methods to look at the chromatin in these patients and look at the transcripts. Why are we doing this? We are doing this to ask a simple question. How is the cell, a neuron in an Alzheimer's brain, different from a neuron in a healthy patient? That's the only question that we are trying to address. So here's the summary of what we find. When we take a mutation, presenilin 1 mutant patient, presenilin 2 mutant patient, APP mutant patient, and late onset patient, patients who are very old and late onset patients, convert the fibroblasts into pluripotent stem cells into differentiated neurons, and do all kinds of sequencing methods and all kinds of approaches to look at the state of the cell, to look at chromatin dynamics. When we do all that, we find that in Alzheimer's patients, there are 4 things that stand out, that are unique, that are really, hallmarks of Alzheimer's disease. The first one is dedifferentiation, which really tells you that the neurons are no longer being neurons. They are going back in lineage to something which is a precursor-like state, which goes back to a cell state which is more stem cell-like than being a neuron. What happens when you have a non-neuronal state? When you have a non-neuronal state, what do neurons do that are unique? They form synapses. So the moment you're not a neuron anymore, you have synaptic function loss, which means that you don't have synapses, you get dementia. What about neuronal lineage repression? In Alzheimer's patients, there's a repression, which means anything that says neuron-like programs are reduced. And these cells, which are Alzheimer's disease patient-derived neurons, they go into cell cycle. You know from your basic, physiology and biology that brain cells they are terminally differentiated. They don't re-enter cell cycle. But in these cases, these cells try to re-enter cell cycle even though they don't finish it. So what are the hallmarks of all of these things? We found that these are the genes that are key contributors to these non-ectodermal differentiation, synaptic function loss, neuronal lineage repression, cell cycle entry. These are the altered gene programs that drive neurodegeneration. So we really went into great detail. I'm going to walk you through the next 4 slides. Very quickly to tell you that this is a summary of what we find. And the way we find it is that we go through this procedure of converting all these cells, pluripotent stem cells, into neurons, 3 weeks of neuronal differentiation, expansion, selection. We get these neurons, we characterize these neurons first, asking the question with the transcriptional, what is common, what is different? If you look at this, for example, there are many programs that are common to all these mutations. There are 730 genes that are common to them, which tells me what is common, what mechanisms are common to all of these. This is where we find out the mechanisms or neuronal specification goes down, dedifferentiation goes up, pluripotency goes up, cell cycle goes up, inflammation goes up. So we uniformly find, using all methods, using transcriptional methods, using transcription factors, the same phenomena is common. Think about it for a minute. You have 3 different or 4 different reasons for your disease. But what happens at the end? At the end, the mechanisms are the same, which means your brain, subject to any type of stress that I'm going to tell you about in just a minute, what does it do? It tries to reprogram itself into going from a neuron into a pre-neuronal state. That means you lose connections, synaptic connections, you get into dementia. So this we find is a new paradigm or new way of thinking about Alzheimer's disease. And so this is the, first time when we show, we in literature showed that Alzheimer's dementia is a cause of dedifferentiation, which means these cells are going back into the precursor-like state. We did the same thing. So this is again a summary of this. The factors that are changing your cell cycle: pluripotency. The pluripotency goes up, cell cycle goes up, dedifferentiation goes up, everything in red. These are key transcription factors that regulate these processes. Neuron specification goes down. In fact, REST is a great modulator of neuronal specification, and that is increased, which means that it represses neuron-like state. And then proliferation and differentiation go down for neurons. So these are all the key processes that are changed. So this is a more beautiful view of looking at all these processes. Those which are red, orange, and yellow are going up, which means cell cycle, immune response, dedifferentiation are going up. Synaptic signaling, neuron lineage is going down. And so these are the factors that are really, I already showed you, these factors are involved in regulating these functions. So now we have a way of looking at the mechanisms that cause the disease. But I have not answered the question to you, how does this mechanism achieved, or what change causes the change in the mechanism? To address this question, before I do that, let me tell you in cell cycle what happens. Why is cell cycle activated? The mutation, for example, in presenilin 1, it alters the Notch signaling pathway. The Notch signaling pathway in normal human beings, you get essentially cyclin D1 is phosphorylated, so it kind of goes into cyclin D1 phosphorylated, which means it doesn't reactivate the cell cycle. Your cells are stuck in the G0 phase, which is what happens in all of us. However, when you don't get phosphorylated, because of the fact your Notch intracellular domain is now activated as a consequence of this mutation, cyclin D1 no longer is phosphorylated. It activates this cell cycle process. It increases the expression of these genes which control cell cycle. You go into G1 to S, S to G2 phase, G2 to M phase. They're all activated, but then it stops here. It doesn't complete the cycle. It is hung around because APC is downregulated, so it's stuck here. So we actually have a really detailed understanding of what happens in this process. We did also experiments involving proteomic experiments to really look at this process to show why the cell cycle is regulated in this way. Let me point out to you what happens. How do we know? What is the mechanism why this happens? So to understand this, we ask the question, how is the chromatin remodeled? How is the nucleosome? How is the chromatin? How are the genes regulated? How is it remodeled? This is a closed chromatin. You know that if you take your DNA in your cell and stretch it out, it's about 2 meters long. How is a 2 meter long DNA condensed into a few, microns dimension nucleus? And the way it's done is it's folded up into all these folded domains. So we ask the question, what is the folded state of this chromatin in a normal brain cell and in a neuronal, and in an Alzheimer's brain cell? So to look at that, we use this methodology called ATAC sequencing, which uses an enzyme called transposase to cleave all the open regions. So when you then sequence all the open regions, what is not open is closed. So you can decipher or infer using computational methods what is open and what is closed. Remember, if it is closed, it cannot express the gene. If it is open, it can express the gene. So there's a relation. It's an alternate way of looking at chromatin topology. So what happens in Alzheimer's brain? So to look at that, we looked at chromatin accessibility using this method. Guess what? I'm going to summarize for you. The key things that are changed are, cell motility, forebrain development, mitotic cell cycle, the chromatin organization. All of this tells you the same story the transcriptome told you, which means that your brain neurons are going to go back into a precursor-like state. To further amplify this, we look at chromatin topology and transcriptional regulation in great detail. This is your chromosome. This is your chromatin fiber. The chromatin fiber is organized by histones, and histones are wrapped around with the DNA. And this is your complete DNA. This is the open region of the DNA. We can look at in active transcription, the closed regions become open. And this K4, one of the regions of histone as K4 gets methylated, H3K4, histone 3 domain, K4 gets methylated, etc. So therefore, we know what marks tell us whether it's open or closed. So we use this methodology that was developed to explore what is open and what is closed. In fact, this is a review we published in Annual Reviews a few years ago. Feel free to look at it. It really provides you with the insight into all these methods. So we then showed that what happens is exactly what we develop. Neuron development is downregulated, neurogenesis is downregulated, neuron differentiation is downregulated, etc. So we were able to show all the downregulated processes. We were able to show all the upregulated processes, which is going back into a precursor-like state. Exactly what I told you before. So when we tried to publish this paper in the beginning, so the journal asked us, what about in real human brain? This is okay in your stem cell model. What about in human brain? So we're very fortunate to get human brain samples from patients who have very similar mutations and transcriptomic data from these. So we looked at the same similar mutation brain data, and guess what? We are so happy to see that exactly the same, this overlap between our work and the human brain, our different mutations in the human brain data showed many common genes upregulated and downregulated. When we analyzed these genes, guess what? We found exactly the same thing. Pluripotency is upregulated, dedifferentiation is upregulated, cell cycle is upregulated, neuronal specification is downregulated. Beautiful result, which tells you that what happens in our model is now happening in the human brain. What does it tell us? What it tells us is, look, this is a great model for studying human brains in a very personalized way. So we can take skin cell from individuals, transform them into neurons, look at the transcriptional programs and the state of the cell in these model systems and see what is the cause. Is there a disease? What is the cause of the disease and so forth? So we now have developed a model system. We showed the mechanisms. Then the next question that you ask is, so what causes it? And guys, what is it? How does it happen? So this is how it happens. The chromatin topology in a brain cell, normal brain cell, your dedifferentiation is closed, not open. Cell cycle is closed, not open. Neuronal precursor maintenance is closed, which means these programs are all, the cell is terminally differentiated. What is open is neuronal differentiation, synaptic function, and so forth. This is a normal brain, non-demented control brain. What about in a demented Alzheimer's brain? Alzheimer's brain, what was closed here? All of these open up. What is open? Closes up. So you're going back into a precursor-like state. And we know exactly cell cycle is upregulated, dedifferentiation genes are upregulated, pluripotency is upregulated. All of these opens up. What closes down? What closes down is everything that's got to do with neuron lineage, neuronal function, and those things are all closed down. And so active programs in a normal brain are now becoming closed or passive or repressed, and repressed programs in the normal brain are now activated in the Alzheimer's brain. So we have now looked at the fundamental mechanisms by which stress in the brain induces this. To further address this question, is it common? Are these mechanisms common in Parkinson's disease? Are these mechanisms common in Huntington's disease? So we looked at Parkinson's disease. We looked at what happens in Parkinson's disease. In Parkinson's disease, the brain regions affected are this region called substantia nigra. And in substantia nigra, you have this protein called misfolded protein called alpha-synuclein. This misfolded protein just aggregates like in Alzheimer's brain. And this aggregated one gives rise to neurodegeneration and Lewy body pathology and other kinds of things. So we said, okay, let's look at this brain, Parkinson's disease. And let's look at Huntington's brain. Huntington's brain happens because there's a region in the brain where there are enlarged ventricles. There's atrophying of the brain, cerebral nerve tissue. And so we said, okay, let's go and look at this brain. What is wrong with it? The way it works is there are these repeats called CAG repeats in the genome, which really gives rise to glutamine. CAG codes for glutamine. So you've got polyglutamates, and these polyglutamates aggregate to form exactly peptides, which are aggregating like plaques, so which gives rise to the same process. So to understand whether the pathology is common across these different brains, a student in my lab, graduate student in my lab, Michael Fitzgerald, he took Parkinson's disease brains, Huntington's disease brain, ALS brains, tried to find what is common across all of these. He looked at the transcriptional programs across all of these. And guess what? When he analyzed the Parkinson's brain, the Alzheimer's brain, Huntington's brain, what is common is chromatin remodeling happens in all of them. Cell cycle is re-entry happens in all of them. Increased inflammation and immune response in all of them. Neuron specification is downregulated in all of them, which means you're no longer behaving like neurons. And then neuron vulnerability increases. And then non-ectoderm lineage, which means neurons come from ectoderm germ layer. Non-ectoderm lineage like EMT goes up. And so this is really a completely common denominator pathology in all these different brains. So our underlying pictorial mechanistic view is the following. Our normal brain, normal stem cells give rise to different germ layers. Ectoderm, which is the germ, one of the germ layers, gives rise to neuronal precursors and forms a neuronal terminal neuron in normal brains. They form microglia, terminal neurons, all these different cell types. In the Alzheimer's brain, you go back in this path, you go back into kind of a non-neuronal state. You no longer have synapses. So you go to a non-neuronal state. It's kind of a no man's land. It is suspended in some kind of a state. It's not a neuron. It doesn't have synapses. It is some kind of a fake cell. And then this cell eventually decays and dies. And then, we get, death. So this is our landscape view of what happens in all diseases, in neurodifferentiation diseases, but most importantly in neurons. So this is our story on what happens in an Alzheimer's disease. Now I'm going to tell you, how do you engineer a drug to deal with this issue? So before I do that, I will again pause for a minute and ask you if you have any questions for me at this point. Do you have any questions for me? I'm hoping you can still hear me.
Yes, we can hear you. We don't have any questions yet.
Okay. I will continue telling you about the story on drugs. I'm going slow, so I'm not going to cover anything much. But that's okay. You can invite me again, so don't worry about it.
Okay, no problem.
So we looked at now therapeutics. How do you design therapeutics? To look at therapeutics, we have a very interesting story I should tell you. So in the 1990s, Eli Lilly, a big company, drug company in the US, they were working very actively on Alzheimer's disease. They developed a drug. This drug is called semaglutide. And they thought this drug was spectacular. And so they did all kinds of animal models. And even though they aren't great, they did all kinds of models. They got FDA approval for doing clinical trials. FDA is Food and Drug Administration in the US. So they got approval for doing clinical studies. So they'd already spent, $15, $20 billion in development. They did these clinical trials in humans, 15 years, 2015. Progressively, they showed that the plaque, through imaging, they showed the plaque in the brain goes down, no problem. But then it turned out that the patients became more demented. The dementia didn't improve. They asked the question, they're very surprised. The plaques are going down, which means the cell, no longer you have occlusion in the brain, but the dementia is not improving. Why is the dementia not improving? They didn't understand. So after billions, $50 billion or something like that in terms of investments, they abandoned the drug in 2015. So because the drug did something, but the drug did not cure Alzheimer's disease. So we asked the question, hey, we have an opportunity to analyze why this drug is not working. And my colleague in UCSD, he had a drug, which is a drug molecule, and it seemed to work better. So what made it work better? And is it good enough? Can we use that as a drug for a clinical trial? So this was the question that we posed. To address this question, we first tested, this is the Eli Lilly drug, semaglutide. And then you can see that this is I'm giving you the structure of the drug. My colleague, Stephen Wagner, he developed a drug, and this drug is called BPN 15606. And he tested both these drugs. In fact, when you test the drug in normal brains, you only have A-beta 38 and 40. When semaglutide was used, it got all of the peptides down, which means that it did not produce even the normal APP processing of peptides that needed to be produced. And whereas in the drug that my colleague developed, only the A beta 40 and 42 were decreased, but A beta 38 and 38 was kept, total levels were maintained. So which means that it only affects the bad peptide, not the good peptide. So first we convinced ourselves that, hey, this seems to work better. But let's see more in detail. So to show what happens in this, we treated the same neurons from these patients and normal brain cells, non-demented control cells, with these two drugs. And then we asked the question, hey, when we treat these drugs, how are the gene programs altered? So when we went to look at this, guess what happens? Both drugs did something in common, which means that they both reduce the cell cycle. They both try to, reduce the process of plaque formation. And plaque formation includes the notion of, essentially not forming, big plaques, and some part of dedifferentiation. But one drug, the gamma secretase inhibitor from Eli Lilly, did worse than the gamma secretase modulator in certain aspects, and I'll show you in a minute. So in fact, I'm not going to go into detail. We looked at all the endotypes in fantastic detail. This is published now, so you can read the paper recently. So you can read the paper. So what I want to summarize for you, what happens in the overall disease in the normal cell, it goes into a differentiated neuron. In the Alzheimer's brain, these are not neurons, these are non-neuronal state. When I treat with Eli Lilly drug, it brings down cells, it goes back to bring down cell cycle, which means represses cell cycle. Notch is an indicator of cell cycle. It addresses to some extent dedifferentiation, because rest is what forms neurons. So it essentially brings down, when rest is up, there's no neurons. So it brings back rest down, brings down cell cycle back. So it's good in that sense. What it does not do is, let's look at what it does not do in a minute. So what about my friend's drug, my colleague's drug? What it does is it does all of these guys. It also does dedifferentiation to some extent, lower it. But neither of them bring it back into a full neuron. Neither of these drugs can bring it to a full neuron. So now we have the ability to test what processes can be addressed by a drug and what cannot be. So we now have a screen. So this new, an engineered screen where we can scale up and test drugs to look at what works and what doesn't work. So this is really the direction we are going in terms of trying to find treatment for Alzheimer's disease. In fact, we are going to try to file a patent which shows that this is the approach you have to take to look at a drug for Alzheimer's disease. Okay, what about a related disease? Let's look at glioblastoma, the brain tumor. What happens in brain tumor? This is
Shankar,
yeah.
Excuse me, Federico. Can I interrupt you for a second?
Please do, yeah.
Before you switch to your next topic. So first of all, we have a question from the audience, and then I also have a question for you before you close up on Alzheimer's. So Brenda is asking if there are any other known causes to Alzheimer's other than the expression of unintended genes. Alzheimer's is a very cruel disease, and I'm always glad to see research done on it, and we all are.
Well, very, very good question. A very good question. Thank you for that question. So let's understand, there can be genetic reasons, there can be, acquired reasons. So this is something well known. The example I showed you was genetic reasons where there's a mutation in the gene, and we looked at what does the mutation do. We followed through the mechanism of the mutation. Why is the mechanism altered in the mutation by the mutant? What happens? That is what we showed by showing differential expression of genes, by showing reprogramming of the cell. Reprogramming the cell gives rise to change the phenotype of the cell. The changed phenotype of the cell gives rise to disease. So this is the train of thought that we have followed. Now, what about people who don't have mutations and still get Alzheimer's disease? And this is called late onset Alzheimer's disease. This is the most common form of disease. So let's look at that origin of this disease. Non-genetic origin. First, we don't know whether there is genetic background in human beings, which can give rise to make some people more susceptible to Alzheimer's disease than other people. We don't know the answer to that. So that's an unknown question. whether somebody's genetic background makes them have a higher propensity, have a higher likelihood for Alzheimer's disease. That's the first question. The second question is what environmental factors or individuals' lifestyle factors can cause Alzheimer's disease or dementia. So I showed you that if you have mutations that's causing it, but you can have stress. The stress can be, we showed, in fact, I'm not going to tell you the story today, we showed in traumatic brain injury, in where you have somebody goes through a car accident and has automobile accident and has a brain injury, or an American football player or a rugby player gets their head bashed in and has a brain injury. These traumatic brain injury has the same mechanistic process. The brains get the neurons get de-differentiated. And this is really emerging as a paradigm for looking at. So what is causing this stress? So it can be toxic stress, it can be oxidative stress, it can be genotoxic stress, metabolic stress. In fact, people tell you certain types of diet can give rise to more common for Alzheimer's disease. They tell you to eat less cholesterol-rich food, this type of food or that type of food. All of these factors are contributing to AD, not just the transcriptional changes or the changes in gene expression or the mechanism by which these stresses manifest themselves into disease. They are not, the transcriptional programs are caused. They are not themselves caused downstream the pathology, but they are caused through other factors before them, and these causal factors or stress factors of various kinds. I hope that answers your question in terms of the etymology or how the disease originates in the first place.
I believe so. I just have to apologize because probably the question came from João Dantas and Brenda just related it. And my question is quite more general, so feel free to postpone maybe your answer to the end if you want to include a more general discussion. But I was really, my attention was really caught by what you show in terms of the general purpose of what happens to the brain. So this reversal to a pre-neuronal, you said, state, precursor state, pre-neuronal. And we're engineers, you made it very clear. These are engineering methods to understand disease. As a more general comment, especially for our students, though, I think that the use that we make of engineering methods, quantitative methods, is also meant to clarify some biological aspects. Now, we're not evolutionary biologists, but we want to answer questions on why a certain disease happens and why it evolves in a certain direction. Otherwise, also the therapeutic design may not be that effective. And the question for you is, why does the brain revert to that? I know, again, it's a very general question, almost philosophical, but what's the advantage in evolutionary terms of the brain going back to a pre-neuronal state? Because Alzheimer's is not an acute disease that you die the next day you reach a certain state, right? So if you revert back to a pre-neuronal state and you start losing functions, in fact, it's a dementia, right? But this doesn't immediately lead to death. So I wonder whether your research, your studies, whether you have a feeling of why this happens, even if this is more of a biology question. Again, our methods, especially for our students, it is good to understand. Bioengineering serves this purpose too. So that's why I'm asking you this question, even if you're not a biologist.
Federico, you asked the most interesting and incisive question that is there to answer. And we have an answer for this. I'm so happy you asked this question. I was going to narrate it after the next 5 minutes, but I'm going to tell you right away because I think I don't want to keep you waiting thinking about it. Let us forget for the moment about brain. Let us look at other processes in human physiology. Just take, for example, your wound healing. If you have a blade that cuts your finger, you essentially rebuild back in going from your, epithelial cells, dermal cells, all these things, they have repair mechanisms and they go back into the native state. You know that. Now, let's take another example. If you look at, damage to your immune system, for example, gets remodeled, continuously produces. Your blood gets continuously reproduced, right? I mean, you know all of these. How does it happen? It happens because when you have damage or stress, your heart is another example. I mean, in fact, people used to, when people have heart attack, what happens to the expression of, you go into more embryonic state when you have a heart attack, right? I mean, this is very well known in literature. Same thing happens in pancreas. You know, when you have cells that are reproduced, you go back, when you have a damage, it goes into a more precursor-like state. And these are all benign. They are good because they are reprogrammed back. So essentially, the cell is sending the message, hey, I'm under stress. I want to save myself. So I want to go back to the more precursor-like state so I can be flexible and plastic and come back to my differentiated state down the road. So we think from a philosophical or evolutionary, not philosophical, from a teleological perspective, when we specify in our chromatin lineage, when I say lineage, we go into different germ layers. We are programmed, the cells are programmed in such a way that under subject to damage, the cells should retreat back into a womb-like state. It's like, I call it going back to the womb hypothesis. So go into a protective state. And this protective state, when the stress becomes even more and more and more, this protective state cannot come back into, it gets more and more into a damaged state and cannot come back into the native-like state. Extreme example of this is cancer. EMT, epithelial mesenchymal transition, is another very good example of this. What is mesenchyme? Mesenchymal cell is a precursor cell, which gives rise to epithelial cells among other cells. So when epithelial cells go into mesenchymal cells, they are going temporarily into a stage, but then once they cannot come back into epithelial cell, they go into tumor. So we have a safety mechanism. The ability to go back under stress to a native-like state is a safety mechanism that is engineered into our chromosome or chromatin biology. When that mechanism fails, which means that essentially the organism has to get rid of the cell, which means that it really kills the cell, right? So when does the mechanism fail? Or it becomes tumor. It's the same result. So when you have a failure of the protection mechanism, so this really happens. In fact, we just published a paper about a month ago on the nexus between life and death of a cell, endothelial cell. In fact, we just published in Frontiers in Cell and Developmental Biology a paper which showed what happens right at the point where the cell decides to live or die or go into senescence. So we showed the mechanisms that decide the fate for the cell. So exactly what happens here, the cell goes into a precursor-like state when it is subject to stress, and to varying degrees. When it goes into a certain form, it says, okay, I cannot go back anymore into my lineage. So I'm going to either go senescent or die. So this is what is our hypothesis. We have evidence for this from multiple physiology, human physiology, from heart, from cardiac systems, from circulatory systems, from lung, from everything. So we believe that this is a universal mechanism that is embedded into all living systems to protect themselves. Federico, did I answer your question?
Absolutely, and thank you very much. I'm sure it was a very interesting and again teleological explanation as well. I just have one more question before
please. Yeah.
You move on from Luisa. Luisa is asking, well, Luisa, first of all, is thanking you for your awesome presentation of bioengineering studies and neuroscience. And then she'd like to ask, what's the difference between metabolic and oxygen distress that you mentioned? So she would like to clarify that item.
I'm sorry, say that again. I lost it. Metabolic.
Sure. The difference between metabolic and oxygen distress.
Okay. Very, very good question. So when you talk about the notion of stress in a cell, stress for a cell or a tissue can be transmitted by many mechanisms. Federico Aletti works on this as well. One of the mechanisms is shear stress, which is that the cell is sheared or subject to some kind of a lateral surface-like stress, which essentially activates channels, which in turn changes the chemistry within the cell. That's one form of stress. When we talk about metabolic stress, metabolic mitochondria is the source of all energy. When you have changes in metabolism, then that gives rise to metabolic stress, which means the amount of ATP you produced is changed. The amount of calcium that is sequestered and changed inside the mitochondria is changed. And so the energy machinery in a cell is changed, and which gives rise to, in broad terms, mitochondria is the one that the recipient of the stress to a large extent. And as a consequence, it changes metabolism. So that's called metabolic stress. In metabolism, oxygen is very heavily involved because there is oxidative metabolism and there is glycolytic machinery, there's oxidative machinery. So when you have stress, you produce, the mitochondria produces free radicals, which are oxygen-free radicals, peroxides and sulfoxides and all these kinds of free radicals. And these oxygen-rich free radicals do havoc. They kind of break up all the proteins. They do all kinds of damage to the cell. In fact, our body produces enzymes like superoxide dismutase and glutathione peroxidase. These are enzymes that counteract the free radicals. The moment free radicals are produced, these enzymes go up so they can reduce the amount of free radicals to get the cell back into living state. Sometimes the free radicals become so high. For example, you take a cell and treat with hydrogen peroxide, high concentration, the cell goes into a shock, various types of oxygen-free radical shock, and the cell dies. So this is exactly what we discussed in our paper. So therefore, the oxidative stress, which can be arising from either hypoxia or hyperoxia, they both give rise to that. For example, when you climb Mount Everest, you have people who die of oxygen starvation because there is your mitochondria tries to act hard to really produce energy and it fails in the end. Or when you go into hypoxia, that's how people die with hypoxia. When you have no oxygen to breathe, you have increased carbon monoxide, but that comes because of the oxidative stress that is induced in the system. Years ago, in fact, we published a paper in my colleague here in UCSD. He brought flies from 21% oxygen, which is our normoxia, normal oxygen we breathe, down to 4% oxygen. He evolved them through generations, for 40 years or so. And then we worked on these flies to see what are the mechanisms that are fundamentally different in these hypoxia mechanisms. And they all have to deal with exactly the question you posed, which is oxidative stress-related mechanisms. So to answer your question, the mitochondria is a source of a lot of these mechanistic changes that happen under different conditions of stress. Did that clear your question?
Absolutely.
Okay.
We have one more question, though.
Yes, of course.
Because questions keep coming. So Jean-Louis is asking, Dr. Shankar, can the formation of beta amyloid be caused by some type of diabetes? And other than being toxic to the organism, can it create some type of mechanical impediment? So I'm not, of course, the recipient of the question, and I'm not an expert, but I do remember, for instance, several years ago when I was a master's student, I almost started a project on amyloidosis and renal disease. And in that case, yes, in the kidney, I know that the amyloids that are formed as a consequence as a cause of amyloidosis do, for example, get stuck in the kidneys, leading to kidney failure. But I guess that, of course, Jean-Louis is asking mostly about the brain and what you presented so far.
This is a brilliant audience here. This is, again, a beautiful question and a very important question. Think for a minute what I told you. When you have a stress in the brain, neurons, this stress induces changes in your cell state, and the changes in cell state give rise to activating the Notch intracellular domain, the gamma secretase mechanism, and gives rise to formation of these plaques. So, we actually monitored this mechanistically very well, right? every cell, many cells produce, all cells have notch activation because notch is a very important pathway. All cells have, presenilin, protein, gamma secretase. When you have stress in those cells, the same mechanisms that happen in neuron happen in these cells. In diabetes, in insulin resistance, where you have the pancreatic beta islet cells undergo dedifferentiation, they go back into precursor-like state. Guess what happens? Same mechanism. When they go into, in fact, there's a beautiful paper, not from my lab, but some other lab in one of the Nature Family journals, in the Cell Metabolism Journal, which showed that pancreatic beta islet cells dedifferentiate in insulin resistance. And they give rise to plaques because that's exactly the problem. The moment the enzyme changes, it does not process APP, which is the normal peptide, by normal mechanisms. It processes it abnormally. The moment you get abnormal beta peptides, you form plaques. The same thing happens in the renal system that Federico was talking about. So these are more common, both the mechanisms of stress-induced changes in the cell state and the ability to go into a precursor-like or lineage state are common across all parts of physiology and not just unique to the brain and to neurons. Okay, I hope that, very, very good question. Very thoughtful question. I hope that answers, Federico, that answers the question.
Yes, thank you, Dr. Shankar. Here is Tatiana. And may I ask you a question? Like, go back a little bit in stress. And
yes.
Dr. Shankar mentioned that are several types of stress that are related to the development of Alzheimer's disease. So you cited metabolic, oxidative, and some physical stress induced by trauma. And And I'd like to know about, have you ever been studying or seen something about physiological stress in relation to Alzheimer's disease?
There is a lot of publications, lot of publications on this area. We don't work in physiological stress alone, but I have no doubt in my mind that, okay, let's look at another facet of stress, which I did not talk about, Tatiana. I should have, this is another area of great interest to us. I'm sure you're familiar with inflammation. So one of the mechanisms by which your body or tissue tries to address stress, and stress can be due to invasion by microbial organisms, right? It can be due to bacteria or virus or so. So the way that the cells address stress is to trigger inflammatory responses. Physiological stresses also trigger inflammatory responses. Very well documented, very well studied area, right? So that's another consequence of stress. So if you're asking whether physiological stress can manifest itself into multiple pathologies, eventually including potentially Alzheimer's disease, the answer is yes. The other main point I didn't mention is that aging is another form of stress, because in aging, your telomeres, which are getting shortened in cell, as a consequence, your ability for the cell to either specify lineage or to multiply becomes lower and lower. As a consequence, more cells die, your tissue, degenerates, essentially you atrophy. And as a consequence, this stress gives rise to even dementia and Alzheimer's disease. Many aged people, get Alzheimer's and dementia, and this physiological stress is induced by aging phenomena. So you're completely right in saying that physiology has a component which introduces stress. And this is one of the reasons why they say do exercise, remain young, eat healthy. All of these things are not just simply TV commercials to make you do a certain specific type of thing, but these things are meant to essentially reduce physiological stress in humans. So thank you for that question. Did I answer your question?
Yes, sure. Thank you. Thank you very much.
Okay, so I want to ask both you and Federico the following question. I'm thinking that we are at 10:41 and I can go for a whole day, but I don't want to. I know you guys have things. So do you want me to say a few more things, or do you want me to stop, or do you want me to, what would you like me to do?
Tatiana, I think we have until 3 o'clock here, which is 11 o'clock in San Diego, correct? So we have another 20 minutes.
Yes.
You're right. We would listen to you for the rest of the day, but I'm sure the other speakers will be also pleased to talk. Tatiana, maybe I suggest that we open the discussion, actually, because there was a number of things we wanted to ask Shankar, right? And it's true, I'm sure he has a lot of things to show us, but I would also take advantage of him, so to speak, to try to interact and talk with him a bit. What do you think?
Sure, sure. It's
Federico,
okay for me.
Yeah, great. Before you do that, let me show you something very exciting for just 2 slides, and then so they'll give you an idea of what happened. I'm going to do some, I'm going to do this movie style, going fast. This is all COVID related stuff. So I'm going to tell you only one thing, that in the very end.
Yes, please. You know, we have a lot of presentations on COVID in the rest. This is a 3-day event. So Tuesday, Wednesday, Thursday. So it's great that you show us something also on that. Yeah. So
we asked a very important question. The question is the following. You know that patients, I mean, people get COVID. Some people recover quickly, less severe. Some people get very severe affliction. They go to hospitals. They get admitted in the hospital. Some of them die. So the general feeling always was, what happens to the immune system in COVID patients? You know, less severe patients and more severe patients. So we did an exhaustive study on a few hundred patients' data on COVID, on the PBMCs, peripheral blood mononuclear cells. And we really looked at these cells to look at how they are different and how there's a remodeling between myeloid system and the lymphoid system. I know many of you know this. Myeloid essentially gives rise to things like macrophages and monocytes and neutrophils, all that. The lymphoid system gives B cells and T cells. And so what happens to the remodeling of the system in healthy patients, in mild affected patients, in severe patients? So we did an exhaustive study. I mean, this is in fact one of the 2 of the Forge papers appeared in print last week. And so in Frontiers in Immunology, this is the key differences between severe and non-severe patients. And we now have a way of looking at the patients beforehand to see whether they will become severe or not. There's evidence of remodeling of monocytes, drastic expansion of the subclass of monocytes called CM1 class. And it happens due to this phenomenon called emergency myelopoiesis. And we know all the mechanisms involved in this myelopoiesis. We know exactly how myelopoiesis happens, and how monopoiesis happens, how myelopoiesis happens. We know about how there is aggregation of the CD14 monocytes. How does it lead to thrombocytopenia and coagulation cascade, and perhaps part of the contribution to severe cases? So I'm sorry. So that's the first phenomenon. Second, we showed drastic expansion of these things called low-density neutrophils. And what is very, very interesting is that this is common to flu and other respiratory disorders, but this is unique to COVID, SARS-CoV-2 infection. This drastic expansion of low-density neutrophils. So we looked at the mechanisms of expansion of low-density neutrophils. Neutrophils are expanding into this because of mechanisms of emperipolesis, which is they go into this megakaryocytes and they have interferon gamma response. As a consequence, they give rise to this special class of neutrophils called PMN neutrophils, MDSCs, myeloid-derived suppressor cells. And these cells are the ones that in turn suppress CD8 T cells and MATE and gamma delta T cells. They are in turn responsible for suppressing it. So this is a crosstalk between the myeloid and lymphoid system. So we showed there is a muted response of interferon response in monocytes and low-density neutrophils, expansion of megakaryocytes, and which means megakaryocytes are the largest cells. They kind of gobble up other cells into them, and that's neutrophils into them. Increased aggregation potential contributing to the pro-coagulative state. In fact, this is what gives rise to blood clotting and coagulation. And then we showed increased netosis, which gives rise to what's called a phenomenon called degranulation. And then impacted the abundance of this myeloid dendritic cells, impaired interleukin-1b response, T cells are suppressed in phenotype. Even the cells are not suppressed. The phenotype is suppressed due to oxidative stress, nitric oxide, nitrosylation stress in the receptor cells. There's expanded killer NK-like cells of one type. There's a change in B cell population. There's a plasmablast expansion, but reduced expansion of naive B cells, which is the one that gives rise to antibodies. So there's a reduced expansion of that. And then the naive B cells become more proliferative, and then they are not effective or active. And there's increased AP1 activity in glycolysis. What about non-severe? More robust response to interferon gamma. Robust antiviral response. Robust antiviral state. High interleukin 1B response. Increased cytotoxic potential, which means that killer cells are not reduced like here. Here, the killer cells are reduced quite dramatically. Robust expansion of adaptive T cell population. Increased OXPHOS, and no distinctive signature to pro-coagulative state. Key point, not pro-coagulative. So we are able to tease out the complete story on the remodeling of the immune system in severe COVID patients using a combination of a lot of engineering methods. I don't have time to tell you. We used a lot of single cell sequencing strategies, analytical strategies, which are involved in very complex methods of dimension reduction and so forth. I don't have time to tell you about that. But I just want to tell you, there's a very latest story from my lab, which really talks about what happens with COVID-19 patients. Well, you ask me, why do you care? You care because we now have 2 drugs. We, meaning my colleagues, have 2 drugs which are now in trials for dealing with severe COVID patients. We now have markers in myeloid and lymphoid system, which we can look at in patients to see, hey, is the patient going to become severe, or is the patient going to be resolved? So these are questions we are looking at. We are looking at post-vaccination patients now. We are not finished that story yet. So we look at post-vaccination patients to see what happens. So this is the kind of, emerging thing in terms of looking at using a combination of methods, the biological methods, engineering methods, and data-driven approaches, quantitative approaches, as Federico called it, to look at disease states. So I will stop now. I don't have time to tell you about how we made muscle, a recent paper, but look, I don't have time. So I'm going to stop here and let you guys ask me questions so that I can answer them.
Okay, so first of all, I really want to thank you, and I'm sure that seeing also the questions that came from our audience, and our students, everyone has been and is excited about your presentation. So thank you very much once again. I will take advantage of the last things you presented. As you know, this is pretty exciting for me. Given my interest in acute disease, I really hope that these data, these approaches that generated these data that are, of course, justified by the moment we have been living in the pandemic, can also be utilized and be useful when we go back to a more normal state, so to speak, and shed light on sepsis in general, because it's evident to me, I've been saying that for a long time, but we've been, bombed by any type of information and misinformation. But it's evident to me that all that we've been saying for years now, that we don't have a clear picture of sepsis and circulatory shock, became dramatic when we dealt with a pandemic of such proportions and ICUs all over the world that were unable to treat patients. So I hope that really, I'm sure that in the future we will also, and hopefully we will together, go back to septic shock, bacterial septic shock, forget a bit about COVID and just advance our knowledge there so we're more prepared therapeutically in the future for such situations. I would like to have another.
Let me pause for a second to tell you that there are many commonalities we draw with sepsis in this paper, with some of the mechanisms that are induced in severe COVID patients. So you may want to look at that too. And I'm very happy to talk to you more about the potential for getting PBMCs from sepsis and then using them to do similar strategies to look at mechanisms that are fundamental to increase septic shock.
Yes, absolutely. Sure. No. And I believe there's much more to understand, for example, about something that you mentioned. So coagulopathies, and you now show the contribution of the immune system We're looking, and other researchers are looking, of course, at other things. But again, I always stress the fact that we didn't know much about coagulopathies in shock in general, and we knew that it happens. And now we had a type of viral septic shock that was particularly conducive to hypercoagulability with the lethal effects that we know. I would like to take advantage of the last few minutes to introduce maybe one or two more general topics of discussion where we all need your opinion. And there are two things that I believe are very interesting for our audience and that you can chime in on with a lot of insight. One is your presentation along the lines that we also mentioned at the beginning clearly showed how important is bioengineering, how crucial it is in a landscape. Medical research is not just about medicine. It is something extremely multi and interdisciplinary. So if you could comment on how we can, as bioengineers, become even more crucial, number one. And number two, since this workshop is dedicated to innovation, what's in your experience? Let's forget a second about some infrastructural issue that's at the core of the differences, for example, between the United States and Brazil. We're not going to get into that. But mentality-wise, as engineers, how can we target, innovation? Besides, a lot of the things you showed are very interesting from the standpoint of even basic knowledge, right? But we always have, in fact, you presented therapeutic design to close the part of your talk on Alzheimer. So we always aim as engineers as practical applications. So I don't have a specific question, but I would like you to comment for our audience. These two aspects: how crucial is bioengineering in this multidisciplinary context, this biomedical research nowadays, and how we as bioengineers, forgetting a second about the external constraints, how we can take care in our vision of this innovation side of our research.
Thank you, Federico. So let me first address the first question in terms of why is now to bring this notion of why bioengineering plays a role. I want to take you back to Leonardo da Vinci. So I like to do this because you get a historical perspective. Leonardo da Vinci, in my opinion, was the first bioengineer. Now you can ask me, why do you think that? So he did the following. He took a bird, a flying bird. He made measurements on the ability wingspan, made measurements on the elevation it can go through, the buoyancy, the pressure that it is exerted to lift it. He did all these measurements, and he in fact designed instruments to make these measurements. So he then took the measurements, converted into a model. Then he converted the model into a design, and he designed the first helicopter. In fact, it's called ornithopter. That's what it's called. And this ornithopter, in fact, is the first. It has not changed. The helicopter model has not changed much from ornithopter. He was the first person to translate measurements into models into design. Right? This is bioengineering at its best action in some level. In fact, Bill Gates bought this beautiful design diagrams of Da Vinci for, hundreds of millions of dollars. I don't know how much he paid, lots of millions of dollars, big zeros. But he has it available now in the museum for you to see that this is how Da Vinci designed it. So what I'm saying is, biology has come to a point where multiple scales are very important. You cannot do only molecular scale. You cannot do only large scale. Gone are the days when you can just simply say that I'm going to take the blood vessel as a tube and simply calculate how blood flows to the tube. Now you've got surface geometry, you've got imaging methods which do all these kinds of things. So clearly, these multi-scale methods penetrated biology, and biologists cannot deal with it because they don't have quantitative methods and tools and measurements to deal with it. So you need engineers to think about it. Now, I can go for long, but I won't go on this area. I want to give you one example where a country like Brazil or a country like India, who I advise, can make dramatic difference in terms of scaling things to doing fantastic research. The area of stem cell biology now is about 20-25 years old. So people know after Yamanaka showed how you can transform a blood or a cell or a skin cell into a stem cell, that part became very, very common. Everybody can do it. But what is the problem? We can all do it in a plate. I mean, we can all do it in a slide or a plate in 2 dimensions. The moment you try to make a stem cell preparation into a 3-dimensional organoid or an organ or a tissue, everything breaks down, because it's no longer a biology problem. It becomes an engineering problem, because you need to look at the stresses, you need to look at the viscosity of the medium, you need to look at the penetration of liquid water, the flow of transport of things, you need to look at thermodynamics of metabolism that is involved. So this becomes a truly engineering problem. This means we need to know the design principles. Just like Da Vinci looked at the design principles of a bird flying, we need to look at the design principles of how our tissues work, and then we can go back and then bring engineering ideas into making a 3-dimensional tissue. And this is now the most important area in tissue engineering. People call about tissue engineering, but this is the most fundamental question. How do we bring engineering concepts towards designing organoids and organs and tissues? And it does not require substantially sophisticated machinery. It can be done in the lab through good ideas, to good thinking, through good models. Da Vinci did not have the latest mass spectrometer. Da Vinci did not have a next-gen sequencing machine, but he was able to design an ornithopter. He didn't have a machining room to do these things, but he was able to do it. So this is really where I think engineering comes to biology. I'm giving you one example. I can give you 10 examples, but this is one example where you can bring engineering to biology, right? Did I make sense, Federico?
Absolutely, absolutely. And I will ask Tatiana if you want to add something, but I think that our students and our audience in general, not only students that are following this talk, loved your mention. I did as an Italian. I loved your mention of Leonardo da Vinci. It was very effective. Tatiana, do we have something more before we close on your end?
We do not have time anymore. We'd love to stay here and talk during the whole afternoon and night, but we don't have any time. So thank you very much, Dr. Shankar, for this awesome presentation. Thank you again for the opportunity to have you here sharing your experience with us, and we are very glad about this. Thank you.
My pleasure, and enjoy the workshop, and I hope I will have an opportunity to visit your beautiful city and meet you all in person. So thank you for inviting me, and enjoy the rest of the workshop.
Okay, it will be a pleasure to receive you here in Brazil in our institute. Thank you very much. Bye.
Bye.
Thank you.
Bye, everyone.
Thank you.